carbon fibers and also hydrogels like that. So basically we modify lignin in the cell walls. And I also work on disease resistance, and all these projects are in poplars. And I mean, these two, the lignin modification and the disease resistance, they are in poplar trees. And you all know that poplar trees, they grow really fast, and we can harvest them in about 3 to 6 years. And it really depends on the end use. So, how many of you have heard about Clemson? If you're a football fan, I bet you know that Clemson Tiger football team are quite famous because they have won 3 national championship titles, and they also make it into to the final round in 2 other occasions. And the genetics and biochemistry programs in my department is also very strong. So I have colleagues who work on plants like um, maize, things like that. I'm sorry, rice, wheat, like that. And we also have colleagues that work on eukaryotic pathogens. And we also have colleagues working in human genetics. And we have this, we have PhD programs, and for the PhD students, they are all fully financially supported, and the stipend, okay, range from 25K a year to 35K a year. It really depends how you are included into the PhD program. And in recent years, actually, it started about 2 years ago, we created these master programs. And for the master programs, it can have several paths. It can be thesis based, or it can be non-thesis based. And for the thesis based, you can do a web lab thesis, or you can just do a literature research based thesis. So I mentioned this is because, as you see here, my name is here. I am the graduate coordinator right now, so I feel obliged to advertise the programs in my department. And there's some misconception that students think some student thinks that they need to have a PhD, they need to have a master degree in order to apply for a PhD program. Actually, that is not the case. Most of our PhD students, they actually just graduate out of college, and many of them actually come from small college, um, like your college. So if you're interested, that's students, for the students, if you're interested or you have questions about the genetics and biochemistry programs in Clemson, just feel free to email me or give me a call. It's fine also. Okay, now that's enough of advertisement. I need to go back to my research. So, like animals, plants, they go through several phases of development. And largely speaking, we can put them into 3 groups, 3 phases. And the first one we call the vegetative phase, and the second one we call the reproductive phase, and the last one we call the senescence phase. So for the vegetative phase, we also call it the juvenile phase, and the length of the juvenility basically on depends on the species. Okay, so it can range from a couple months in annual plants to several decades in coniferous species. and and lots of things um, can control the regulation of the reproductive phase when the flowering start. The flowering actually signals the onset of the reproduction phase. And all these phases are important because they are part of the plant's life cycle. But for reproductive phase, they are particularly important because it produces offspring, right? And so it has a lot to do with the survival of the plants. And through sexual reproduction, the genetic variations are introduced. So it has a lot to do with the evolution also. And it also have a lot to do with our food and food security, and it has economic importance. Just imagine this. Okay, so lots of fruits we are eating, and the rice and the flour and some of the vegetables like cauliflower and broccolis, they are all part of they are the result, they are the product of the sexual reproduction. So without the sexual reproduction, we would not have this stuff on our table. And in real world, there's a need to regulate the flowering in plants. For example, you are a seed orchard manager, and your trees takes several decades to flower. So to set seeds, so you do not want to wait in those 20 decades and then without any income, right? Everyone wants to make money, so you do not want you want to get seeds, you want to get products from your seed orchard. And also in for fruits, okay, they have this phenomenon called the alternate bearing. So this year you may have a heavy harvest, but then the next year you might have a light harvest or nothing at all. So we can regulate this fruit setting, basically it's the flowering, to mitigate the impacts of the light year. We call the light year. Okay, and sometimes we also need to ensure uniform flowering. For example, if you have exhibit coming on and you want your plants to flower so that you can have a nice display, right? Or you are a nursery owner and you have these many ornamental plants and you do not want them to flower at once because you might not be able to sell them all in once. So you might need to allocate some plants and you treat them first. You made them flower first, and so that you can sell this set of plants, and then you come back and make the second set of flower, and then you can sell the second set. So in the real world, there's a need to regulate flowering. And through model annual plants, I'm talking about Arabidopsis here, we know that there are 5 major flowering pathways. Okay, the first one is called the Photoperiod Pathway. So means the light cycle regulate the flowering time. And then there is this mineralization pathway. So that is the temperature, the cold temperature, it regulates the flowering time. And then we also have this autonomous pathway. Basically, this pathway is independent of the light and of the temperature. And then we also have this gibberellin pathway, and we also have the H pathway. And all these pathways basically, okay. All these pathways actually convey the signals to these, we call it the floral pathway integrators. So these central floral genes, and then they induce the flowering. So these genes include the leafy gene, the FLY gene, and the AP1 gene, and SOC gene, and also include FT genes. So all these 5 pathways, they convey these signals, these informations to these central genes, and then induce flowering in Arabidopsis. And so this is a simplified picture of the flowering regulation in Arabidopsis. And in reality, there's other elements also, like other hormones. Besides the GA, there's other hormones, for example, the auxins. They can induce flowering in some plant species also. And the nutrients of the plants, especially the carbon to the nitrogen ratio, and it has a lot to do with the flowering time. So we can do girdling or root trimming to induce flowering. And of course, stresses like a drought can induce flowering also. But how these other factors, these elements get involved all these pathways, currently we are not that clear. Okay, so I give one example here. This is girdling. So basically you peel off a layer of the bark from the trunk, and what it does is um, it decreases the nutrients in the plants. So the plants kind of starved of the nutrients. And this kind of stresses can induce flowering. And some of you may already know that the reproductive growth competes with and reduce vegetative growth. So if the plant grow really rigorously, they usually do not flower. They just keep growing, okay, and they would not flower. But on the other hand, if the plant flower too much, you got too many floral buds in here, and now you do not have leaves. Okay, you do not leaves in here. So when the reproductive growth is too much, it can inhibit the vegetative growth, and sometimes it can kill the plants. So this is a plant from my greenhouse. At the beginning, when I saw that in this tiny plants, I got 3 flower buds in here, and I was so excited. But later on, the plant just died. It's not because of drought. I assure you that. It just, the plant do not have enough nutrients, and they just die off. So I'm going to give you a couple examples that I have done to induce flowering in woody plants. The first one is in Metasequoia cryptostroboides. Oh, it's a mouthful. So I just call it Metasequoia because this is the only species in the genus. And in US here is known as the Don redwood, and it takes about 25 years to set seed. And this is species, the fossil record show that it existed as many as 50, oh my, million years ago. But it was only discovered in 1947. So you can imagine that when this species was discovered, seeds were collected from the site, and then they create these seed orchards because they want to propagate this species, right? These tree species. But it takes about 25 years to set cones. This is a coniferous species, so I use the word cones. So what I did is that use this a combination of the auxins, oh, the IBA and the zeatin to spray on the leaf or on the shoots, and it is quite effective in producing the male buds and also the female buds. Okay, so it's quite effective, and with this hormone treatment, I can reduce the cone setting time from 25 years to 5 years. So no matter it's grafting plants or seedlings, the, the treatment, the hormone treatment is effective in 5 year old. And as you see here, for the plants that are younger than 5 year old, the hormone treatment didn't work. So this underscores the importance of the vegetative growth before the reproduction. And the second example is golden camellia. I think you might not have seen this golden flowering camellia before because they are very, very rare. And the common colors we see are pink, white, or red, and sometimes purple. Okay, but this golden color, um, yellow flowering camellia, they are very rare and they are very expensive to buy. Okay, and its flowers contains lots of antioxidants, so we use it for tea also because it can improve health. So a tiny plant that's about 65 centimeters, it costs about $70 per plant, and for flower teas, it costs $120 per 100 gram. Is dry tea, so it's dry. So there's a need to induce early flowering in camellia, and in the wild, without intervention, it takes about 6 to 8 years to 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 flower, and at the early years of flowering, they only produce a couple flowers. So um, it just a need to induce early flowering for uh, economic reason. So for this species called a Camelina nitidissima, okay, I use this growth retardant chemical called the pacrobutrazole. So another mouthful again. So I just call it PBZ. And this one I did not spray on the leaves because it can cause deforestation. And so I put it into the root. Okay, just put it into the pot. Apply the solution, the PBZ, into the pods. So for this species, we are able to induce flowering in 4-year-old grafting and also 4-year-old seedling plants. And as you can see here, for higher concentration, this is different type of concentration of the PPZ. For higher concentration, you can induce close to 100 floral buds, okay? But for lower concentration, you still can get about 20 floral buds per plant. So that is there's a benefit to this. First, you cut the juvenility time from 6 to 8 years to 4 years, and then you can produce lots of flowers. Okay, and this year, actually the experiments started last year, but now we started to get a result. So we apply the same PPZ to another golden camellia species. So this species is native to Vietnam, and the flowers are bigger, okay, and the leaves also are bigger also, and it's also very effective. And it for the 120 for the 100 and 200 ppm concentration, we can induce about 20 flowers per plant. So from these two case studies, what did we learn? First, we know that different hormones may work for different species. So for the Metasequoia, we use auxins, and for the golden camellia, we use a PBZ, a growth retardant. So for different species, you might need to try different hormones because they might have different effects. And secondly, the treatment times is very critical. Okay, you basically, when you do this whole this flowering induction, you need to do lots of sectioning to look at when the buds begin to differentiate. Okay, you need to catch the right time for the treatment. For Metasequoia, it has male and female buds on the same trees, but on different branches. And we noticed that early treatment around late May and June, the same hormone solution induce male buds. Okay, and then for treatments that are done in later of the summer, like late July, early August, it induce female buds. So it's very critical that you know when the floral buds start to initiate, to differentiate. And the third thing we learned from these two case studies is that higher concentrations of hormones may have better effects. When I say the better effects, means it may produce more floral buds, but it has some side effects. So you've seen this photo before. So this one, we actually used a higher concentration, and then we have all the leaves pretty gone. And then the second year, the plant died. Okay, because no leaves, no photosynthesis, no nutrients, so the plant died. And we in this table you can see that so this data is for the severity of deforestation. Okay, so for the higher concentration of the PPZ, the plants, they drop leaves prematurely. Okay, but for a lower concentration, the leaves they do not fall that easily. Okay, and um, and when you have this severe defoliation, it actually affects the plant basal diameter, the stem basal diameter. So this is the severe defoliation, and you can see the stems are smaller. And for plant height, um, it it we did not see a significant difference for this study. And the fourth thing we learned from this case study is that the hormone treatment can have a sustainable effect. What I mean is that this year you made a treatment, and the next year you see the flowers, and then the following year without treatment, you still can see some effect. You still can see some flowering occurred. Okay, so this is what we learned from the case study. And now you might wonder, okay, you induce the flowers, you induce the seeds, how they perform compared to the naturally occurring ones. So we did the dissectioning to look at the microspores and the megaspores in Metasequoia, and morphologically, we did not see difference between the induced one and the naturally occurring ones. And the seeds, the pollens can germinate normally, and the seeds can germinate normally also. And we have the healthy seedling growing from the induced seeds. And for the golden camellia, because like I said before, we vary golden camellia not just for the beautiful flowers, we also use it for tomato, right? So we check the active chemical components in the induced flowers and compare it to the naturally occurring ones. The naturally occurring one is from the 35-year-old trees. So these, the induced one is from the 4-year-old plants. As you can see here, this is some difference, but they're not that dramatic difference. For example, for the polysaccharides, so this is the naturally occurring flower one. So it has a higher content of the polysaccharides, and the induced one a bit lower. But for saponins, actually the induced one have a higher level, and for the polyphenols, here is the naturally occurring ones. The 200 and 300 ppm treatments have similar levels, and for the flavonoids, the induced one at least have similar level as the naturally occurring ones. And again, the naturally occurring one, the naturally occurring flowers are from the 35-year-old trees. So those flowers, the natural occurring flowers are bigger, okay? So the induced one are smaller in terms of diameters and fresh weight. But when we compare the dry weight, actually the difference is not significant. So actually the induced one is a bit heavier in terms of dry weight, but again, the difference is not significant. And now we want to understand how the hormone treatments induce flowering, right? The mechanisms of the action of the hormone treatments. And you all know that endogenous hormones play important role in development. So we look at the IAA and the GA and also the ABA in the metasequoia treated plants. And we also look at the the hormones in the golden camellias. So from both studies, we can see that the hormone treatments actually alter the metabolisms of endogenous hormones. So we can see the difference after the treatments. And let's look at this one. So in metasequoia, you can see that the higher concentration of the GA and then the lower concentration of the IAA and also the BAP actually is beneficial to the male bud initiation. Yeah, I just noticed I got a misspelling words in here. And for the female bud, actually, it's you have this higher IAA, okay, and then higher GA, and then the lower level of the ABA are favorable to male cone initiation. And in golden camellia, you see this is the induced one. Okay, the red color is the induced one, and the blue color is the control untreated ones. And you can see after the PBZ treatment, the IAA level didn't change much during the floral initiation, and but the ABA increase, the ABA level increase, and also the GA level increase, and but similar to the IAA, the SA, okay, the SA level actually is lower, okay, especially around this time when the flowers start to initiate. Its level is lower than the untreated plants. So from here, basically we know that the hormone treatments from outside, they actually moderate the metabolisms of all sorts of hormones inside the plants to regulate flowering. And another way to say it is that the various hormones, they actually cross-talk to regulate the flowering transition. It's not just one hormone do the only work. So basically is the various hormones work together to regulate the transitions from the vegetative growth to the reproductive growth. For the metasequoia work, we also look at the non-structural carbohydrates, and you can see that this I think this is not hard to understand for this one, because flowering, it takes energy. So you can see the glucose all go down in the flowering shoots or the cone setting shoots. Okay, so in the treated shoots, you have a lower level of the carbohydrates. In the molecular level, we want to understand how the floral genes are regulated after the hormone treatments. So the first step we did is create a floral um, transcriptome for Metasequoia. And we identified 59 floral gene family genes, including agamas and AP2, the constants, and leafy genes like that. And you you saw the simplify. You saw this figure before. So basically, all these major pathways, okay, they involve different genes, and most of these genes homologs were identified in the Metasequoia transcriptome, the floral transcriptome. And this tell us that these central genes, they may have similar functions across different species between the gymnosperm and the angiosperm. And in Metasequoia, we also identify two very interesting genes that may be involved in the floral bud transition. So one is called the AGL24, and as you can see here, that is the transition. Okay, that's the transition from the vegetative bud to the male bud. Okay, and the second one is the AGL19, and this one may be involved in the transition from the vegetative bud to the female bud. Okay, so this clustering analysis clearly suggests that these two genes might be involved in the male and female bud transition from the vegetative growth. And the database also identify lots of hormone signal genes, such as the ABC transporter family genes. Okay, and we also clone some of the central genes, because the central gene basically that's the integrator homologs from Metasequoia and overexpressed in Arabidopsis. The reason we did it in Arabidopsis is that, as you know, Metasequoia is a coniferous species and it's a very difficult to do transformation. So we expressed the FT gene and also the leafy gene into Arabidopsis, and the transformed plants actually flower early. Okay, flower early than the wild type. Okay, so this is the leafy mutant, the Arabidopsis mutant. So, but the leafy gene and the FT gene from Metasequoia or induce flowering early. So the two examples I just gave you is try to induce flowering, right? You want to plants to grow to flower early, right? But for some cases, you do not want flowering to occur. So if you you are allergic to pollens like I am, so now these days I cannot do my exercise outdoors basically because I'm really allergic to pollens. So in this case, you might want to reduce flowering in the landscaping plants like that. And in China, these species Platanus is a popular tree species in China. I think Legion must know these trees because you're from Shanghai, Nanjing area. So when these trees start to set seeds, they form this we call the birds, right? BURS, and they will drop down from the trees and hit people. Of course, they won't hit people badly, but still, it's not a pleasant experience. So we try to reduce flowering in this species, and the way we did it was spray them with GA, gibberellin, because gibberellin that is a very important hormone, but it has different effects in different species. For example, in Arabidopsis, GA promote flowering. So GA treated Arabidopsis flower earlier. But in fruit trees, like mangoes and apples, when you treat them with gibberellin GA, the fruit trees will set fruits less. Okay, so we use GA to spray the trees or along the streets, or we can just inject the solution to the trunk. And the next year, you can see that there's less fruit on the trees. So this is the untreated one. You can see lots of fruits in here, but the treated one, there's much less. So flowering induction or flowering inhibition, they all have their applications in the real world. For the future research, if I find the money, of course, that is the condition. I want to look at the global gene expression after the treatment. Okay, so basically, I would like to use RNA sequencing to look at what genes, what networks, what pathways have been altered after the hormone treatment. And then I would like to use them to look at the metabolisms of the secondary compounds, especially hormones. So that's two major directions of research I would like to go to. Now I have a small grant from the South Carolina Department of Agriculture to continue to work on golden camellia. And I'm trying to introduce new species to US, okay? Because in US, we do not have this golden camellia. And I try to introduce the new species and then try to improve the clonal propagation for the golden camellia. Okay, now I need to tell you why I chose this research project to tell you about, because it is quite close to my heart. I started this project when I was a graduate student, when I was doing my master's degree back in China. And my advisor was Professor Weilen Yin from Beijing Forestry University. And as I told you before, a metasequoia, it takes about 25 years to set cones, to set seeds. And when this species was rediscovered in 1947, seeds were collected from the remaining trees, and then lots of seed orchards were established. But they just did not set seeds because it take 25 years. So my advisor sent me to this province, to Hubei province, and work on this project. And it was really an adventure for me. I stay in the farm, the tree farm, alone by myself, and prepare the solutions, collect the data, and make the treatments. And sometimes I need to visit tree farms in very in the mountains and did the treatment. So it was a bit scary, but when I think back right now, it actually I really cherish those experience. And when I came to US, I got a grant to continue to work on this project, and it's USDA grant and is an international collaboration. So later on, I worked with Professor Cindy Xia in Beijing Forestry University, and then the then graduate student Ying, she came to my lab and spent a year in my lab to continue to work on the project. And for the Golden Camellia project, I work with Professor Jinlin Ma and Professor Xiao Jianwei from Guangxi Forestry Research Institute. And for the Plantanis project to inhibit the flowering, I work with Professor Zhao Huatang from Shanghai City Landscape Management Bureau. And that's what I have for today. And thank you for listening. And if you guys have questions, please let me know.
Thank you. That was wonderful. Yay.
Thank you.
Okay, we have a couple of questions. Let me find my chat again. Okay, sorry, hang on one second. So the first question, I think you answered a little bit of this. Any long-term impact on induced early flowering plants?
Yeah, some of the plants, they continue to flower at least in the second year or the third year after the treatment. And after that, I didn't have data for continuing to flower. And some of the plants, the one that were treated with a higher concentration. So for Golden Camellia, we started a project in 2019. I just gave away my age. We started in 2016. So we didn't know what concentration would work. So we tried a wide range of concentration from 50 ppm to 750. And then for the higher concentration, and some of the plant die after they flower. So they die in the third year.
Mm-hmm.
And for the Metasequoia, the trees actually, for the 5-year-old tree, actually is pretty big. It's about more than one meter high. So they did not die. Those plants survive. They perform better. That's what I try to say. And as you see in those photos, after my experiments, they actually do mass spray on older trees because they have big sea orchids. They are at least 10 years old.
How do you decide which hormone to use for which plant?
Okay, so we try a few things at the beginning, but at the beginning we need to do lots of literature search and see what might work better with the coniferous species and what might work with hardwood species, and we narrow it down and then try them out.
In the past, I heard that causing stress to a fruit plant by hammering a nail to its roots causes them to produce fruit. Is that the same as girdling and ring barking? Does
Yes.
it have the same effect?
Similar mechanism. So if you have a plant that grow beautifully, okay, in terms of vegetative growth, they usually do not flower. And another way to say that you can, if you want your plants to flower, you cannot treat them too well.
You have to make them suffer a little bit.
Yeah, but there's a threshold, like the tiny plants I showed you. They flower a lot. Tiny plants, they flower. They got 3 floral buds, and eventually it just kill it.
Mm. To introduce golden camellia to the U.S., what kind of regulations do you need to go through?
Okay, so in USDA, U.S. Department of Agriculture, they have this office called APHIS. I don't know what APHIS stands for, but it's more like a regulation office. So they regulate your transgenic plants also. So for example, I grow transgenic trees in the field, so I need to go through these applications to get a permit. And the same thing for importing plants. You need to have a permit, and there are all kinds of import permits, and you need to go through all these descriptions. But my suggestion is that you just email them to get a quick answer. And because my experience is that the first one I thought this is the right one, and at the end my plant got killed at USDA in Atlanta. The office is there. They say, sorry, you didn't have the right permit, and it took me 2 years to get a plant to travel from China to US, and then it got killed in Atlanta. And but those people, they are super nice. They will tell you you should apply for this and that, and what's the difference between those permits. So I would suggest a quick way is to call them up or email them and get better ideas instead of what I did before, just waste time and energy.
Can you explain super briefly what grafting is, and did you see a difference in the grafted plants and the seedlings?
Okay, you mean how does the grafting works?
Yeah,
Okay.
some folks may not know about grafting.
Right. Yeah. I forgot about that. For grafting, you have a root stock. So you have a plant. Usually it's from seedlings, but it came from cutting also. But no matter what, you have a plant that already have a very nice root system. And the root stock usually they can be drought resistant, or it can be soil-borne pathogen resistance, like or microbes, including like nematodes. So the root stock, it can provide several benefits. So can be drought resistance and disease resistance. And so you have this root stock, you cut the top off, and then you open, you just slide slide the stem of the root stock vertically, and then you put your plant insert into that cut side, and then you perifilm them and let them grow. The condition is that these two plants, they need to be related. So you cannot graft an apple tree into, let me say dramatically, say poplar trees.
Hmm.
So they need to be, it can be your apple and pear. They because they are in the same genus. I have not done this before, but at least they need to be related. And the more related they are, the more successful you will get. And for seedling, we use this word to generally describe the plants that grow from seeds, that germinate from seeds. So for seedling, you have a uniform genotype.
Mhm.
For grafting, the root stock is one genotype, and then the top one we call the scion. The scion is a different genotype.
And did you see a difference in the treatments from
For the
seedling and
treatment,
grafting?
okay, you see the seedlings, when I say, for example, when I say 4-year-olds, seedlings and cuttings, the 4-year-old seedling usually is more juvenile. The cutting is more mature because the cutting is from a plant that has been that are already growing.
Right.
For seedling, for 4-year-old seedling, you start from 0. And for 4-year-old cutting, actually they have some growth already. But for the treatment effects, you will see better effects on cuttings. Another way to say is that if you can get an effect on the seedlings, I can say that you would not have problem to have effects, the flowering effects from the cuttings. Oh, graftings, I'm sorry, graftings.
Right.
Yeah.
Thank you. Another question, what is the difference between research use versus commercialization?
Research use. You mean for the techniques?
I don't know, do you want to clarify your question? Or regulation?
The regulation? For flowering induction, I don't think USDA imposed any regulation on it, unless the chemicals we use is very harmful. Then in that case, when you do the spray or you apply the treatment, you might need to comply with certain regulations. Yes, that's not something I'm familiar with. In my greenhouse, nobody's tell me that you cannot do the treatment.
And the hormones that are sprayed on the trees, those don't affect humans, right? The plant hormones, or do they?
Okay, so for the metasequoia, they're hormones, right? So whenever we deal with the hormones, you need to wear gloves and you need to wear mask. Even though you just like you buy the hormone, like rooting hormone or the flowering promoting hormone from Amazon, everyone can buy that, but they will have a warning tell you that you need to wear gloves, and if it touch your skin, how many washes you need to do. And if the hormone come in powder, it is suggested to wear a mask because it's hormone. It can induce growth or inhibit growth.
Yeah.
It depends on what type of growth or hormone it is.
But not in people, in plants. You're talking about spreading it from plant to plant? Or does it affect people?
You mean when do the spraying and in
Yeah, so if you're spraying and say I breathe that in, is there a problem with that because it's the plant hormone? Is it different enough?
even though it's plant hormone, yeah, you still need to wear a mask. And in US here, not a regulation, just they suggest you to change your clothes if you do mask spraying. In my case, since I use a handheld sprayer, a handheld bottle, so I just need to wear a mask.
Excellent. Any other questions? Wonderful. Well, thank you so much.
It's my pleasure, and it's good to see. I'm sorry I couldn't see all of you, but like I say, if you're interested in my graduate program,
Yep.
feel free to contact me. And if you want to do a summer internship, feel free to contact me also.
Yes, students, if you're listening, summer internships. So anybody that's interested can contact her directly or contact me, and I'll get you in contact with her. I'm getting a lot of thank yous and good job on the chats for you. So I think it was really informative, and I appreciate your time, and I think everybody appreciates your time. And thank you so much for doing this for us.
Thank you for the invitation, and it's nice to know some of you.
I'm getting a whole slew of thank yous and that they enjoyed it coming up in the chat. So everyone seemed to enjoy it.
Thank you.
Well, thank you everybody for joining us, and I will be sending out an invitation for the next one, which is coming up in April. So thanks everyone for coming, and I hope everyone has a good night.
You too, thanks.
Thank
Good
you.
night.
Bye. Thank you.
I thank
Good
you.
night.
Good night.
Bye.
Good night.
Thank you so much.
And Bernie showing up.
Good to see you, Haiying. So long. It's beautiful work, very impressive.
Thank you.
Congratulations. Yes, actually, I never learned all this. So thanks for the opportunity.
Yeah, I didn't do this part when we were together.
I know it's new, exciting. Thank you. It's so good to catch up.
Yeah,
we see each other, but then
right. Okay. See you guys, and thank you, Regina, for the invitation again.
Thank you so much for your time. I really appreciate it, and the students appreciate it.
Okay.
Good to see you. All right, I'm gonna shut off now. Thank you.
All
We'll be
right.
in touch.
Bye.
We'll stay in touch. Okay.
Yep. Bye bye.
Okay. Bye.